Good evening, everyone. My name is Em, and I work at the Welcome Genome campus. And tonight is our session, Genomics Lite, and we're looking at the human genome in context with Karen McLaren, who is our guest speaker for tonight. So we've also got Fran here, who also works for the Welcome Genome campus, and we'll be sort of fielding questions and helping with anything as the session goes on. You'll see, as you've probably all used Zoom webinar before, but we've got the chat box. So if you've got any general questions for us or any tech issues, pop them there and me and Fran will try and help you out. If you've got any questions for Karen, you can either put those in the chat box as we go, or you can use the Q&A function. Either's fine, whichever one you'd fancy. We'll be using polls at a couple of points during the workshop, so they'll just sort of pop up on your screen, and if anyone can't see them, we'll also read out the question and you can use the chat box as well. So I'm going to give a very short introduction on the campus before we hand over to Karen. So let me just share my screen. Awesome. Can you all see that all right? Fran or Karen, can you just confirm that that's all good?
That is good.
Awesome. So all three of us work at the Welcome Genome campus, although I started during lockdown, so I've actually not visited campus yet. But in normal times, this is where we would be. We're located just outside of Cambridge, and on the campus we've got quite a lot of different scientific activities happening. So on the campus there are two main research institutes. So we've got the Wellcome Sanger Institute, which is where Karen works, and we've got EMBL EBI, which is a biodata and bioinformatics building. So EMBL EBI focuses solely on dry lab, on sort of computer analysis of biodata, whereas the Wellcome Sanger Institute has a mixture of sort of hands-on wet lab and computer analyzing as well. To give you a little bit of sort of background and context to the campus, I'm going to play just a really short video just to give you a little bit of background on the campus.
Greenwich, London, site of 0 degrees longitude. Geneva, Switzerland, home city of the CERN Large Hadron Collider. Just as the Greenwich Observatory is considered the home of time, and CERN the heart of nuclear physics, so this campus on the edge of Cambridge is forever linked with the science of genomics. The Wellcome Genome Campus is where the first animal genome of a simple nematode worm was sequenced in the late 1990s. And now the campus is leading a revolution in genomics and biodata, bringing the prospect of previously unimagined medical breakthroughs. So, scientists from all over the world are attracted to work here. Let's see how they define what a genome is.
I would say the genome is a blueprint of all the information needed to generate a whole organism.
Every organism has its own genome.
Yeah.
Just as the genome connects us with all other humans, you know, we all share much more of our code than we have that is different.
Not only between each species is the genome different, but actually each individual of that species has a slightly different genome.
It's those instructions that define how the natural world looks.
That is much better than I can explain.
Awesome. So that's just to give you a little bit of background on sort of what the campus is and the sort of work that we do, but Karen will also go into obviously much more detail than that. Just before I hand over to Karen, just want to point you towards yourgenome.org. So you'll see on the website that you signed up for this webinar on, we've got linked resources specifically for this webinar series, but on the yourgenome.org website, there's lots of resources, videos, activities about anything related to genomics. So if you're looking for anything for revising or for learning more about DNA, that is the place. I will stop sharing screen, and then I will pass over to Karen, who will be giving our talk for this evening. If you've got questions, feel free to pop them in the chat as we go along. I might interrupt Karen if they're sort of pertinent to one bit, or we'll just leave some of them for the end as well. So Karen, I'll hand over to you.
Absolutely fine. Thank you, Em.
Right, let me just get the slideshow working. Let me swap. Can you just see the slide and not the presenter view? Right, well done me, got it right this time. Thank you very much. And a big thank you to everybody for joining us. I know it's quite late in the day, getting dark outside, but hopefully I'm going to give you a bit of background on the original Human Genome Project. We're going to do some history and go back to that point, and then hopefully I'll bring that right forward to the present to show how that original work is the backbone of what we do today. So to kick off, I wanted to give you my CV. So my journey through Sanger, at which I've worked there for over 26 years now. So a little bit about my background, and then a bit about what I'm doing now.
Sorry, Karen, I think that your PowerPoint is sort of slightly zoomed in. We can only see sort of the top quarter of the slide.
Ooh, funky. Right, let me have one moment.
Might just be worth coming out and going back into it.
Yeah, let's do that. Can you see my presenter one now?
Yeah.
Right, let me swap that again.
Yeah, it's weirdly zoomed in.
Oh. Right, let me stop the share and then reshare. See, this is the joy of the Zoom thing, isn't it? Right, let me try going full screen on that. Share. Right, let's give it another whirl. Of course, when the computer's ready. Right, you can see. How about now?
Yay. Yeah, much better. Well done.
Awesome. Normally this used to flummox us, but actually after being on Zoom since March, this is part of the course. Right, let's get back to where we were. Slide still normal? Yeah, fab.
Yeah, all good.
All right. So yeah, me and my journey through Sanger. Now in that intro video that Em played you saw the nematode worm, and that was my first introduction to genomics. So working on the sequencing of that, and we're going to talk a little bit more about that because that's the thing that catapults the Sanger into the Human Genome Project. Obviously worked on the Human Genome Project, going to talk to you about that. And then I went on to be involved in some of the other model genomes, including the mouse and the zebrafish, which we undertook solely at Sanger. So the first part of my career at Sanger was very much around the genome projects, but actually the latter sort of half, I guess, has been project managing. So I draw on my experience of the organisation, my background in science, and data to manage projects. I've got a, as you can see, there's a large diversity there. I work on setting up systems, refurbing and kitting out new labs. One of the things, one highlight from there that I'm proud of is the work on the IPS lab. So an IPS is an induced pluripotent cell. And these are cells, you can take skin cells, you can reprogram them to be undifferentiated and then redifferentiate them. So this is one of the exciting new aspects to our science, among lots of other things. So that's that's me now, project managing. But let's start with the history. So a question before we dive into some of the black and white photos. So do you want to pop the first poll up?
Awesome. So there should be a poll jumping up on your screen somewhere. We're just sort of generally trying to ask if you're familiar with the structure of DNA. If you can't see the poll for whatever reason, feel free to pop your answer in the chat. But it looks like most people have been able to answer. Most are saying yes, got 1 or 2 no's. Give another couple seconds for people to have a chance to vote. Awesome. Looks like most people have had a go at voting. Most people have said yes, but there's a couple of no's amongst there as well.
Cool. Okay. So I'm happy then if it sounds like most people are familiar. So, but if I cover something and anybody wants any clarity, yeah, stick a question in chat or Q&A. So that's me. There we go. Okay, so this is the history part. So we're going to think back to 1950s. The gentleman that you can see on your screen is Jim Watson and Francis Crick. And here they are pointing at one of their models to show the structure of DNA. So the twisted ladder, the double helix, and the bases that form the rungs of the ladder. This photograph here is one of the models that sits, this is part of the Science Museum collection, one of their original models. The story about the discovery of the structure would certainly not be complete without looking at the work also of this lady. This is Rosalind Franklin. So she, along with her colleagues, were part of lots of the discovery work. The image you can see here is actually an X-ray crystallography image. And from this, she was able to interpret the physical structure of the molecule. So although in much of the literature, Watson and Crick are credited, it's a team effort, and posthumously the recognition has been given more greatly to Rosalind. So this that you can see here, so I'm sat about 2 miles away from the centre of Cambridge in my home. And actually, if you went into the centre of Cambridge, you'd see that one of our pubs has a blue plaque on the wall. And this is the pub that Watson and Crick burst into in 1953 to say that they discovered the secret of life. So when they elucidated what the structure looked like, they thought the best thing to do was to go to the pub and announce that. And in 1953 then, so off the back of this, they write a paper telling the scientific world about the structure, showing, as you can see in the diagram, the shape. Interestingly, this extract from the paper talks about the bases, but it doesn't, obviously at this point, the order of the bases, that's the piece that's not understood. But this is really where people credit the story beginning. So if we move forward in time a little bit, this gentleman is Fred Sanger. So as you know, I'm working at Wellcome Sanger Institute. We take our name from Fred Sanger. So, and this is to do with the fact that he pioneered sequencing methods. So how could we find out what the bases were along the strand of DNA? Fred Sanger is actually one of only a few people to have been awarded more than one Nobel laureate. And the one that resonates most with us at Sanger, that gives us the name, is the one that he jointly won in 1980. And this was about his contribution to how to determine those bases. And the two images that you can see here show the use of the Sanger technique and how it became developed. At the top image, so the bases used to be labelled with radioisotopes, and then we could read them on the outputs. Obviously, working with radiation is not ideal. And more latterly, so Fred and his colleagues looked at replacing those radioisotopes with fluorescent labels, so much safer to work with. And it was this discovery and advancement that really kick-started the idea that maybe we could sequence a human genome. So as I said, it was really that excitement, that possibility from the technology that got the scientists really excited. So if you rolled the clock back to 1980, when Fred achieved that Nobel Prize, actually, if we'd have actually undertaken the sequence then at the speed that you could sequence at, it would have taken us about 600 years. But that did not put off the scientists, so that made them more hungry to do this and to push on and try and make the sequencing faster. I've just seen a question come in. How do we read the sequence from the graph? Did you? So if I've gauged the question correctly, in this one, each base is in its own lane, and you read from left to right. Usually you would go down the way. You know which base is in which lane, and then you go through them in order. So if this is A, then there'd be another A, a C, a G, etc. But with the fluorescent labels, we can actually bring them to life with these colors. So, and you can see that the colors are matched to the peaks. I hope I've answered that question correctly, but feel free to ask again or get clarification if you would like. So let's, so we've got scientists super excited about doing the Human Genome Project. Even though it might take them 600 years, they're pushing on, they're trying to develop the technology. And actually within a decade, so by 1990, the possibility of sequencing a human genome was really viable. And the speed of sequencing had developed such that we estimated it was going to take us 15 years. And so then the Human Genome Project was born. So in England, this gentleman here, this is Sir John Sulston, and he was instrumental in the UK at getting funding so we could set up sequencing here in the UK and to take part in the Human Genome Project. So he doggedly sought this funding. You'll see in the background, there's that nematode worm again. So John had physically studied the nematode worm, so he was really au fait with the biology, but then he went on to do the sequencing. And that was a proof in principle, that kind of trailblaze that if we could work out exactly how to approach and do it for the nematode, then that would really help us with the human genome. Did I see a...
Yeah, there's just a second question, I think, as a sort of follow-up to that first one. So asking what the number at the base of each peak was. So I think if you go back,
oh yes,
maybe
cool.
2 slides. I think it was on this slide, but I can't see any numbers. So,
But typically, if I hadn't cropped the image, there would be numbers here. And what the numbers would do is you'll get a number to give you a probability for the base that it's showing you being correct. So these, when you have these really clean peaks and they're very distinguished, not many overlaps, you'll get a really high probability number. And actually, when they're like this, slightly overlapping and smaller, the probability value would be lower. So I hope I've answered that one correctly, but awesome. Love the questions. So let's go back to John and his very scientist looking haircut. So he's lobbied Wellcome and MRC to get enough money to begin the Sanger Centre. So we're now Wellcome Sanger Institute, but at its very start, this is what John put together. And if we look in on the Sanger Centre as it was then, by 1993, we're sequencing John Sawston's nematode worm. We're also doing some yeast genome sequencing. And we are beginning to map out and do the planning for how we're going to attack our chunk of the human genome. So you'll see that the aerial shot here looks very different. We moved into these premises that belong to an engineering company. So they were not fitted out as labs, but such was the appetite to get started. We made things work. And you'll see now that our campus has got some fantastic purpose-built buildings. So, as I said, we're going to talk a little bit more about the worm, and it was the proof of principle that we could. It taught us lots about how we should approach the Human Genome Project. And in 1998, we published the complete sequence of the nematode worm in the journal Science. Now, when we did the initial analysis, at that point, there was somewhere in the region of about 20,500 genes in the nematode genome. So I think on the next slide, we've got another poll coming up. So hold that number in your heads, that 20,500 genes for a nematode. So this is a microscopic soil living worm. Now, we're starting to do the Human Genome Project alongside. And actually, this picture here is a notebook that Ewan Burney, now one of our joint directors of EMBL EBI, that Em referenced at the start. He opened a book with everybody at the Cold Spring Harbour meeting, where you could pay $1 and guess how many genes the Human Genome Project, the human genome, would have. Now you'll see these numbers around the outside. These are the guesses from the heads of each of the labs. But Em, if you pop the poll up, we'd like you to have a guess too. So various numbers on there. So have a go.
I think we've got votes for every category so far. So people think
cool.
that it could be really small or huge. I think currently the most popular answer is over 200,000 genes, although.
Sure. And you'll see actually that's fairly consistent with what the lab heads were thinking also. So the answer is actually somewhere around 26,000. Now that number changes slightly. So this was a big shock to us working on the genome projects. It seemed incredible that we only had a few thousand more genes than a microscopic soil living worm. And this was one of the big findings from that Human Genome Project. And it really sort of taught us that it's not how many genes you've got, but it's what you do with them that counts. Fab. So let's look at the human genome. So the image here is a karyotype of a genome. So we refer to a full genome as diploid, meaning there are 2 of each chromosome. So we've got 22 pairs of autosomes, and we have our pair of sex chromosomes. So we can tell something about this individual just from looking at the karyotype. We can see the X and Y chromosome, so we can identify this as a male. We can actually also see that there are 3 copies of chromosome 21. This is known as trisomy 21, and it's this additional copy that's responsible for Down syndrome. So even at this very high level, we can tell something, but obviously what everybody wants to know is to get right down in there with the detail and see what the sequence is telling us. So when we talk about the Human Genome Project, we are talking about the haploid genome. So we only sequence 1 strand of one of the pairs of chromosomes. And here we have the size of the human genome. So we're looking at 3,000 million or 3 billion base pairs. Often this is referred to as gigabases, or sometimes we just use the GB. The nematode worm actually was 100 megabases in size, so much smaller, but obviously with lots of genes. The image that you see here, if you could get to and obviously access the Wellcome collection down in Euston Road, there was actually a printout of the entire human genome, and it fills a bookcase floor to ceiling, just giving us a bit of a sense of scale about how much information is actually there. So lots of bases, and as we know, it was going to take 15 years. It was going to be very expensive as well, and so the principle that the original Human Genome Project gave us was this divide and conquer. So typically in science, at that point, scientists would be very guarded on their research. They wanted to be first to publish, but the Human Genome Project really gave us this concept of actually, if we divide up a very big problem, we're going to actually achieve it. And you'll see when you look at our research today, especially around big data, this is a thread that we carry the whole way through at Sanger. So we're super proud. This is John that we saw with his nematode worm heading up the lab at that point. And we were responsible for sequencing and curating 33% of the genome. So the biggest slice of pie out of all of the labs involved. This was important because it really put Sanger on the map in the UK. And what we saw is as we did more of this work, we started to get more scientists wanting to come and work at Sanger. So it was a real attraction factor and putting our reputation up. So this slide here, so on the left, I've no idea how old everybody is in the audience. If you're as old as me, you'll recognize this is a piece of acetate. So all of the big heads of all of the labs got together in Bermuda to talk about the data that was being generated and think about, should we have any rules around that? Now, the key things that came out of that meeting was that they decided that yes, all sequence should be freely available, and actually it should be deposited for the public to pick up on a daily basis. So this is a really important central concept. So at this point in time, to look at parts of the human genome sequence emerging, all you needed was a web browser. So nothing more than that, and this really opened up the possibilities to both researchers that's sitting at their bench, but also, you know, big companies looking at disease, etc. And this principle is upheld today. Most of the large scientific journals will have an open data policy, meaning if you conduct an experiment using sequence data, but you do not make that data available so somebody could reproduce the experiment, they will often turn down the submission. So this is a really key point for what comes after. And again, this is the seed for what is now part of our research, which is big data. So our sequencing back in 1993, 94 was done on slab gels. I don't know if you're familiar with running small electrophoresis experiments as part of your curriculum. We essentially, you've got two A3 pieces of glass with some gel in between them, and then we load the DNA and we draw it through the gel using electricity and those fluorescent dyes that we've attached. We point a laser at them and that excites them, and then they shine, and we record the color and that probability value. So a concept that is important to note. So you cannot sequence currently from one end of a chromosome to another. So all sequencing techniques will require you to break the DNA into smaller fragments. So there's that. And also, none of the sequencing technologies then, and indeed now, are 100% accurate on all bases all the time. And the way that we deal with that is that we oversample. So rather than just sequencing every base once, we sequence it a number of times, and then we're looking for the consensus, and that gives us a better confidence of the base that we're calling is the correct one. So you've got lots of small bits, and you've got to put them all back together. This was a computational challenge because we didn't have an original genome as a backbone. So the race, let's talk very briefly about this and its part in the Human Genome Project. The gentleman that you can see there is Craig Venter. He was originally part of the public effort for sequencing, but then in 1998 he declared he'd started a company and he was going to sequence all of the human genome. He was going to do it faster than the public labs were doing, and he was going to put that data into a database, which would be a pay to search. So you would not be able to sit there with your PC and see the genome emerging. You would have to pay the company some money to review. So at this point, we've invested a lot of work and a lot of money in the human genome. And what this did, I don't think this was the intention of Craig at that point, but actually it galvanized our efforts. So everybody, all of the funding bodies put more money into it, and we decided we would just work extra hard and we would flood the market with freely available sequence. So he did not get traction with that product. So that whole thing about let's flood the market with this free data, it really galvanized all of the technology companies as well. They could see the whole project gathering pace, and that actually we were going to do this thing, and so lots of technology companies popped up, and they carry on driving sequencing technology so it's quicker, more efficient, and less error prone. And that's part of that change in pace that comes about with Craig Venter's announcement. So by the middle of June 2000. So you remember we started in 1990. We predicted it was going to take us 15 years, but actually within 10 years, we put together the first draft of the human genome. Now it's got holes at this point. There are gaps, which we'll go on to work on later, but we declare that there's enough of the sequence there that you can represent the genome of a human being. We had global press announcements, really exciting time. This was our Prime Minister at the time, Tony Blair. This is Bill Clinton, President of the US at that point. And this is Craig Venter, who actually came to join our press conference to say how pleased he was that we'd achieved the milestone. So really, really exciting time. That in that decade we've gone from what looked like a crazy idea to a reality. Now, if you look online, there is a Nature Collections paper. I think Fran has got the links for this, Em. I don't know if you have these to pop into the chat. There's lots of great articles in here. It's about the actual chromosomes themselves, but it's also about the work and the labs involved. And I would endorse what Em said at the start, which is the Your Genome site has got some fantastic resources on there. Lots of the questions that I'm covering here, additional things. So I would definitely encourage people to have a look at Your Genome. So this is the team that did the human genome at Sanger. Imagine the size of this team and all those labs all around the globe that are taking part. Certainly, it takes a lot of people to do something this significant. Interestingly, whenever the media came to film us, they didn't want to see all these people. They wanted to have a picture of any robots or just some funny coloured liquids. And we all generally had to get out of the way to give it that sort of slightly sci-fi feel. So I would heartily recommend this book. This is co-written by John Sulston and Georgina Ferry, called The Common Thread, and it really is it's a very easy read. And actually it talks, there's lots more detail there about the science and the politics, but also, and this is something that John Sulston was very, very keen on, is exploring the ethics around doing that sequencing and what it would reveal. And I guess the best quote from the book in terms of when we got to the end of the Human Genome Project was like John's comment that actually we're at the beginning. So the minute we could sequence one human genome, that's not enough. Actually, then people wanted to sequence more. The scientists started having really amazing, exciting questions about, if we sequence lots of individuals, what will we find out? So, but definitely recommend that book. In that as well, John talks about he broke his hip at one point and had some meetings with our funding body in London, which he still attended after popping some painkillers in. So such was the appetite to just get this thing over the line. So let's just reflect then on, so we got our 3 gigabases of sequence, but the Human Genome Project gave us some other valuable legacies as well. The strength in numbers, that strong international collaboration, as I say, not a typical thing then in science, but absolutely is now. And Sanger, both Sanger and EBI are all about those big data, providing that connection and getting lots of minds involved in solving problems. So we see that today. Free access to data, as I say, the world, I'm sure, and the Sanger, I'm certain would look extremely different if the data had gone in pay-to-view databases. And it made everybody look at sequencing, but also data storage, and think about how we can store lots of data in a smaller space. So I'll just touch very briefly on that note with the bioinformatics side. So this is what we refer to as bringing the computer thing together with the biology. And our data center on site has somewhere in excess now of 70 petabytes worth of data storage. Your PCs at home, if they're high spec, you may have a terabyte. So it's a huge amount of data. I've got a figure there for 1 petabyte. Actually, if you were playing MP3s, that's about 2,000 years worth. And this just I think demonstrates the scale of the amount of data. And this is our precious product. And this is what we work with EBI to look after and to share with the world. So we're going to have another poll. So one of the things, we're bringing it forward now. We're landing it into 2020. So the cost of sequencing has come down. So there's two questions. One is, how much do you think that original Human Genome Project cost? And then, so we've got some guesses on there. So have a go. It's all in dollars. But just interested on your thoughts. How much do you think it cost?
Generally quite a lot by the looks of things. Quite a lot of votes for sort of 100 million and higher. Quite a lot of votes for between 500 million and 1 billion, and a couple of votes for over 1 billion. No one's going for the cheapest option, so I think people at least think that it's a fairly expensive thing to do.
And that's exactly right. So the cost estimates are in excess of $1 billion. Somewhere between 1 and 2 billion. So an expensive genome for sure, which is why actually we divided the whole thing up between various labs in the world. So the time to sequence a genome has dropped dramatically as well. So the original human genome, the one that cost over $1 billion, that took us 10 years. Emma, I can't remember if we've got another poll here or
not for this one. We've just got one more
cool.
to have at the end.
Perfect. So the time to sequence a genome now has actually dropped from 10 years to 2 to 3 days. So for the same quality of product, a phenomenal shift. We've got
a really interesting question come up about which human genome did you sequence, in that, like, whose was it? Was it from a volunteer or was it from an established human cell line? So very good question.
Yeah, awesome question. And so the answer is the donors are anonymous. Those of us working at the time, we had our suspicions. And I say donors because the original human genome has actually got 6 people who donated DNA. So at that point, the techniques for harvesting enough DNA to go on to do the sequencing at the quality we needed were just not there. And so it's a mosaic of 6 different individuals. The ideal would have been to do 1. So it's 6 anonymous people put together, but fabulous question. So our data yield, and this is the link here, that you can now get more sequence out of a smaller amount of DNA. You needed loads when we start off, but now the amount needed is so small, we can collect samples, clinical samples. So we can take a few cells or even a single cell and sequence the genome from that. So current sequencing technology is mind-blowingly faster. We are churning out millions of bases per second, and this is all day, every day. This generally goes on 365 days of the year also. So the sequencing technique, so we still use Fred Sanger's concept of the fluorescent dyes, but the way that that is now sequenced is different. It takes place on a super small space, something like a microscope slide, so yeah, sorry, I was just checking the chat. Bye Fran. Also, so we're now looking at lots of data coming from a very small space, but also a good quality genome is now sub $1,000. And so that's a massive shift. And when the scientists see that cost coming down, they want to do more. Let's sequence more genomes and find out more, ask more scientific questions. And just flick my cursor back here. This is one of the newer sequencing machines. They are, the footprint's big, but again, you're basically putting 2 very small cartridges in and getting back an enormous amount of really good quality sequence. So let's just to finish then, just to pull it right forward to today. So this is our campus today. So we're no longer in those buildings that we moved into. We're in state-of-the-art, built for scientific research laboratories. And so research on campus. Now I'm literally just going to whiz through these. I would encourage everybody to have a look at the Sanger website, which brings all of these to life, and the Your Genome. But all of our research programs that sit around human health and human disease, such as parasites and microbes, for example, they study malaria. But because of the technology, they study both the malaria genome itself, the mosquito genome, and also the human genomes of those who are severely affected and also not. And that's part of enriching the amount of information we've got for drugs to control that. Our biodata we've mentioned lots, and we've very recently included in our suite of research a project called Tree of Life, which is actually ambitiously attempting over a period of time to sequence every species on Earth, something which would not have been possible if we hadn't nailed those original genomes. So I can't remember.
Yeah, we've got one more poll, so I can pop that up.
Yeah, and then we've got a question.
Awesome. So we're, yeah, the poll on screen. We're interested to know whether you would be interested in having your genome sequenced now that it's much more accessible than maybe it used to be.
And there are no right or wrong answers here. For me, this would be a no. I don't want to know. I think I'm quite content to just leave it to leave it to my lifespan to get to. What are your thoughts?
I think if I had a good sort of medical reason to think it might be useful. So if, for example, I had a relative diagnosed with something that was partly genetic, and I had a chance of getting it. So if one of my parents was diagnosed with something that was genetic, I think I might be inclined to for that reason. But without that incentive, I think I'm happy not knowing for now. But it's definitely an interesting one, and I think that looks like a lot of people feel similar. So we've got quite a lot of yeses, but we've also got a lot of maybes, and so I feel like it's probably people in a similar thought process, that there's definitely pros, but there's also cons, and you can't unknow once you know.
I think that's it there. I was just flicking to the question there. how are the number of genes counted? So the gene count is done using a process of looking at the sequence called gene annotation. And so they build on information that they've discovered from organisms where the biology is really well known, i.e. the nematode. You look for signatures in the DNA, and from there they do the gene count. Now, at the end of the Human Genome Project and with the nematode, that was manual curation. So we had a huge team of people going through, looking for different sets of bases, signatures, checking those with other bits of information. It's automated now, but yeah, at the time it was a long process, hence we finished sequencing the nematode genome, but it actually takes us a year or so to properly count all the genes and publish.
But a lot of scientists are very pleased that it is automated. Doesn't sound like the funnest task.
It doesn't, but I guess it was, you know, that's the groundwork too, and then the people could then go on to train the algorithms to do it automatically. So I'm going to end there, and as Em said in the chat, please, any questions, so happy to take those. I think probably the key thing to end on is say, for me, it's a pleasure to represent Sanger, and it's thanks to the scientists we've got on the support teams. However, I think the human genome story really shows us about those scientists with those crazy visions who just didn't stop pursuing them, were the people that really made it all possible. So I will end it there. And
there's one more question that's come up, a couple of questions that come up in the Q&A. I'm aware of what the time is, so if anyone does need to sort of leave us now, then a recording of this will be made available in the next few days. But those of you who'd like to stay and listen to these questions, then obviously you're more than welcome to. So someone has asked, what are the cons of having your genome sequenced?
So it really depends on the personal situation. I mean, I think, so one of the things is, because we know that there are particular genes which they don't make it certain that a particular disease will occur, but they certainly tell you that your chances are very much in that way. One of the other things is to consider, once your genome is out there, other members of your family can then infer from your genome partly their genetic predisposition. So I think that's one of the things, and this is what John tried to promote along with the sequencing, is once it's out there, it's out there. And whatever story that tells, whether that dictates that you're very likely to suffer from dementia or not, for me, I think that's the con. I think, and very personally, I think if I knew too much, I'd be so worried. I probably wouldn't. I probably wouldn't be able to live my life and enjoy myself as much. But quite I guess the answers are quite personal there.
Yeah, I think the sort of the pros and cons around finding out about genetic predisposition is whether or not there's anything you can do about it.
Yeah.
If you find out that you're more likely than average to get something like Alzheimer's or dementia, where at the moment there's not an awful lot that medically can be done, then it's whether or not you want to live with that information. I think it's often different for things like breast cancer, I think is the sort of common one that people think about, where if you've got a family member that has it, you've got a higher chance of having that specific gene, but actually there's quite a lot of things that can be done about that. So in some instances, knowing about your predisposition means you can take action, but in other ones it means that you can't, there's nothing that you can be done. You just have to live with that information. So I think it's, yeah, as Karen said, it's a personal whether you want to know that about your biology or not.
Yeah, but it's a great discussion to have, because there are no right wrong answers. So another question, did we think the sequence in different individuals will be the same as you use 6 people's DNA in the initial draft. Let me just, so I think that question then, so it's asking about, I believe that mosaic effect.
Yeah, I think so.
So the answer was, when we started, we didn't know how different individuals were going to be, and actually as we went along we realized, so when we sequence a whole chunk of human genome from one individual, and then if we're sequencing a chunk next to it from a different individual, we would have an overlap. So there would be a bit that we'd sequence from each individual to help string it together. And actually when we started doing that, we realized how many differences there were, and we would expect to see in 1,000 bases, we expected to see at least one base that was different between individuals. Now that's in the general parts of the genome. There are parts of the human genome, such as areas on chromosome 6 that looks after immunity, etc., where the rate is much, much higher. And so in those cases, when we try and put overlaps together, we have to say, well, do these go together? Is this just a lot of differences, or are these supposed to be apart and there should be a piece in the middle? So it's something that we discovered as we went along. And when we sequenced the zebrafish genome, because that's more than one fish, we see a really high rate. So it was an even bigger puzzle to, is that 2 fish that look different or not? So yeah, about one SNP, one single nucleotide change in 1,000 bases, and we didn't know that before we started. The only way to find out was to sequence. That's a great question.
Awesome. And I think that looks like all the questions for now. So thank you very much for everyone coming along. If you think of any questions after you leave our session, you can email us at the email address that's on the website that you use to sign up. That'll come to myself and Fran, and we can ask Karen for any extra questions that come along in the next couple of days. But yeah, thank you very much for coming along. The registration for the next 2 sessions, the next 2 weeks of sessions, are now live on our website, so do check those out. Next week we will be talking specifically about new sequencing techniques, and it's quite impressive how far it's come in a relatively short amount of time. And the week after we'll be looking at lots of different careers in science as well. So thank you very much for coming along today. Thank you very much, Karen. And yeah, thank you very much.
Superb. Thank you, everybody. Have a good evening.